Good day, this is Professor Will, CBMD PhD. Today is Wednesday, July 17, 2019. It's 13:18:42 seconds Eastern Standard Time. And this is a continuation of my scientific reading on pathophysiology. So let's get started here. Multiple injection safety violations found in New Jersey septic arthritis outbreak. Tainted knee injections, infected 41 patients caused $5 million in Medicare claims. Date July 17, 2019. Source: Society for Healthcare Epidemiology of America. Summary: multiple violations of injection safety and infection prevention practices, from lack of hand washing to inappropriate reuse of medication vials, were identified after an outbreak of septic arthritis at a New Jersey outpatient facility in 2017. According to a recent investigation, investigators found 41 patients with osteoarthritis contracted the rare painful infection following injections in their knee joints, including 33 who required surgical removal of damaged tissue. This large, costly outbreak highlights the serious consequences that can occur when healthcare providers do not follow infection prevention recommendations. Following initial reports of 3 cases from a local hospital to state and local health departments and multiple complaints directly to the facility in March of 2017, the facility voluntarily stopped performing procedures. A state infection prevention assessment team identified 41 cases along with multiple breaches of recommended infection prevention practices, including inadequate hand hygiene, unsafe injection practices, and poor cleaning and disinfection processes. After collecting medical records and self-reported data, the team of medical and public health professionals from state and county health departments, along with the New Jersey Division of Consumer Affairs, conducted an unannounced visit to the facility. The site visit included staff interviews, reviews of medical records, evaluation of regulated medical waste handling, and observation of mock procedures performed by the staff while the facility was closed. The assessment team found multiple breaches of proper infection prevention and injection safety practices, including the lack of hand washing stations or alcohol-based rub, and the exam rooms, exposed syringes with injectable substances drawn up to 4 days in advance, and inappropriate handling and reuse of single-use and multiple-dose vials. In addition, exam tables where injections occurred were cleaned at most once a day, while surface cleaning prior to each preparation is recommended unless a clean barrier is used. Nationally recommended infection prevention and control practices are applicable to all settings in which health care is provided. However, outpatient settings sometimes fail to provide the infrastructure and resources needed to support infection prevention activities, and often lack regulatory oversight. Before the facility reopened, state officials provided recommendations from the Centers for Disease Control and Prevention's 2016 Guide to Infection Prevention for Outpatient Settings. Minimum expectations for safe care. An infection prevention consultant was recommended to review practices and assist with changes. No additional cases were reported after the prevention recommendations were implemented. Further research is needed to identify how to best provide infection and prevention training and education to healthcare providers in outpatient settings, researchers say. Infection prevention training should be emphasized at all levels of professional training for all healthcare personnel upon hire and on an ongoing basis, with oversight to ensure competency. Outbreaks related to unsafe injection practices indicate that certain healthcare personnel are either unaware, do not understand, or do not adhere to basic principles of infection prevention and aseptic techniques, confirming a need for education and thorough implementation of infection prevention recommendations. Good day, this is Professor Will, CBMD PhD. Today's Wednesday, July 17, 2019. It's 13:47:42 seconds Eastern Standard Time. And this is a continuation of my scientific research on pathophysiology. So let's hop to it here. New insight into microRNA function can give gene therapy a boost. Date July 17, 2019. Source: University of Eastern Finland. Summary: scientists have shown that small RNA molecules occurring naturally in cells, i.e. microRNAs, are also abundant in cell nuclei. Previously, microRNAs were mainly thought to be found in cytoplasm. The scientists also discovered that microRNAs concentrations in cell nuclei change as a result of hypoxia. The findings strongly suggest that microRNAs play a role in the expression of genes in the cell nucleus. The scientists profiled the division of microRNAs in different parts of endothelial cells, discovering that a large share of microRNAs are enriched in cell nuclei. When the scientists exposed a cell culture to hypoxia, they noticed that changes in the concentration of individual microRNAs mostly took place either in cytoplasm or in the cell nucleus. This profiling study is the first one of its kind, showing that microRNAs play a more essential role in regulating the expression of genes in cells than previously thought. For instance, the scientists found that microRNA 210, a molecule previously strongly associated with hypoxia, is in fact especially abundant in the cell nucleus. This observation sheds light on previously unknown mechanisms that cells use to adapt to hypoxia. MicroRNAs weaken the expression of their target genes by binding to the ends of their messenger RNAs in the cytoplasm. This phenomenon is known as RNA interference, the discovery of whose mechanisms was recognized with a Nobel Prize in 2006. Researchers showed already 10 years ago that synthetic microRNA molecules can regulate genes of therapeutic importance in animal models by targeting their impact on genes' regulatory areas and cell nuclei. This discovery, along with subsequent research, led the scientists to assume that structurally similar microRNAs occurring naturally in cells also play a role in the regulation of genes in the cell nucleus. It is highly significant that these microRNAs targeting the cell nucleus can also increase the expression of genes, which is opposite to what happens in RNA interference. This is a very important finding in view of novel gene therapy for researchers. Good day, this is Professor Will, CBMD, PhD. Today's Wednesday, July 17, 2019. It's 14:10 and 37 seconds Eastern Standard Time. And this is a continuation of my scientific research on pathophysiology, so let's hop to it here. How kissing as a risk factor may explain the high global incidence of gonorrhea. Date July 17, 2019. Source Monash University. Summary. In 2016, there were 87 million people diagnosed with gonorrhea, the most antibiotic resistance of all the STI. There is a global rise in gonorrhea rates, and until now, no one has understood why. Kissing as a major risk factor for gonorrhea is a hot topic in the field of sexually transmitted diseases. A study published earlier this year by the research team studied more than 3,600 men who have sex with men over a 12-month period from March 2016. By mapping those who only kiss partners compared to having sex with partners, the researchers were able to determine that the transmission of the disease is high in people who kiss only and was higher in those who have sex with kissing compared to those who have sex without kissing. Researchers argue that the global sexual health community needs to recognize that gonorrhea is on the rise and that there should be an increased awareness of the risk of kissing as a route of transmission. Understanding how it is transmitted is the key to understanding how to control it. If transmission by kissing is a key route of transmission, then it is important to investigate new methods of control such as antibacterial mouthwash. Good day, this is Professor Will, CBMD, PhD. Today's Wednesday, July 17, 2019. It's 14:54 and 57 seconds Eastern Standard Time. And this is a continuation of my scientific research in pathophysiology. So let's hop to it here. Living longer or healthier. Genetic discovery in worms suggests they can be separated. Date July 17, 2019. Source University of Pittsburgh. Summary. Gene identified in worms controls how resources are allocated for stress resilience, longevity, and fertility. Aging research indicates that better health span, the quality of life as we age, may be more important than lifespan. Researchers suggest that there may be molecular switches that control lifespan and health span separately. Health span is represented by a set of parameters like mobility and immune resistance that are distinct from lifespan, which can be easily measured. Though it is harder to study, in the long run, it may be more relevant to modify health span. In the current study, the research team focused on a protein called TCER1 in the worm Caenorhabditis elegans. Earlier work from their lab showed that TCER1 promotes longevity in worms and also is critical to its fertility. Longevity genes in many animals increase resistance to stressors such as infection, so the researchers expected that removing TCER1 would make the worms less resilient. Much to their surprise, they saw the expected opposite. When infected with bacteria subjected to DNA-damaging radiation or high temperatures, worms without TCER1 survived much longer than normal worms. They also had improved mobility with age and were less prone to protein clumping that causes human neurodegenerative diseases. Conversely, increasing TCER1 levels beyond normal suppressed the animal's immune defenses. A researcher was sure they made a mistake somewhere, but that researcher repeated the experiments and realized that TCER1 was unlike any other longevity gene they'd seen before. It was actually suppressing immune resistance. Interestingly, TCER1 seemed to be able to wield its influence only as long as the animals were young and capable of laying eggs. I liken TCER1 and C. elegans to a DJ who controls the bass, treble, and other tones to get the music to sound just right, says the researcher. During its reproductive age, TCER1 tunes all the molecular dials to ensure that the animals reproduce efficiently to propagate the species, partly by diverging resources meant for stress management. Researchers caution that it is too soon to make any conclusions about human health span, but notes that the findings should change how we understand the molecular basis of aging. It will be interesting to understand how the body allocates resources. Researchers speculate. For example, could women one day take a pill once they decide to stop having children that would improve their health span by diverting resources used for reproduction toward improved stress resilience? Good day, this is Professor Wills, CBMD PhD. Today is Wednesday, July 17, 2019. It is 15:33 and 15 seconds Eastern Standard Time. And this concludes my scientific research on pathophysiology. Hope you enjoyed listening. Have a great day.